Next speaker is Robert Kallal, and it's a great title, Tales of Schizomid Tails, Patterns in Schizomid Flagellum Shape from Elliptical Fourier Analysis. Anytime you're ready, Bob.
Great, thank you, Mike. So today I'm going to talk to you a little bit about schizomid flagella. This is work I collaborated with my advisor, Hannah Wood, and Gustavo Miranda, who exposed me to these arachnids, and this will actually be the first talk I've given not on spiders. So schizomids are the sister lineage to Thalaphonids, and Thalaphonids of the two are probably the better known of the groups. Vinegaroons are another name for Thalaphonids, and they are well known for this defensive spray, kind of this elongate, annulated flagellum. And the females of schizomids also have a similar flagellum, but today I want to talk about the males. And the male flagellum is a little bit different. And here it is on this photo by Marshall. So the flagellum function has been explored by many authors, and recently by Monjarras Ruedas and friends, and Ruiz y Valente. And what happens is during the mating march of schizomids, the female will grasp the male's flagellum, and then he will position her over his spermatophore. And the flagellum shape is important to, it's thought to be sexually selected, of course, and is important to ID and diagnose these groups. So much has gone into homologizing the CD and the annuli and the lobes and depressions on the flagellum of the male in particular. So what I wanted to do as part of this study is to quantify flagellum shape and describe the major axes of variation in this male schizomid flagellum, test for differences in disparity, as well as testing for allometric relationships. I wanted to look at land area versus disparity and diversity correlations, and finally look at the taxonomic utility of shape for diagnosing different schizomid genera. So the male flagellum has long been understood to be important for diagnosing schizomids, and for that reason it features in the majority of their descriptions. For this study, we generated our outlines for our morphometric analyses from the primary literature, and this is extremely useful in times like these, where access to institutions are limited. But these outlines can vary in quality and type, what they show, what they don't show. They might be illustrations, they might be photos. And here's some examples. So to look at the shape difference, we elected to use Fourier-based morphometrics to summarize shape by curves rather than landmarks. So if you saw Erica's talk earlier today, she used this technique as well. Essentially, you are introducing more curves or harmonics to give an increasingly nuanced outline of a given shape. And these Fourier coefficients can then be input for PCAs and other multivariate analyses. We conducted these analyses in the R package MOMOX. So this type of technique has been used in other arachnid groups, such as spiders, Apiliones, Amblypygi, and recently by Erica, her Solifugi. And how this works is, here's an example flagellum of Saurozomus kitu, and the kind of gray-blue shape is the outline with increasing numbers of Fourier harmonics. And you can see as more are introduced, the shape is captured better and better. So as part of the study, we wanted to look at the dorsal view and the lateral view of the male schizomid flagellum. We got 283 outlines in the dorsal view, and for lateral we have 129 and counting. So for now we are going to focus on the dorsal view. So these outlines were centered, aligned, and scaled for analyses. And here are the PC loadings for the principal components analyses that many of the downstream analyses are predicated on. So you can see that PC1 explains 72% of the data or of the variance, PC2 about 13, PC3 6, and PC4 about 2%. So most of the variation is essentially long and slender versus broad, followed by the relative length of the pedicel and where the widest part of the flagellum is. Then the relative width of the pedicel compared to the rest of the flagellum, as well as the location of lateral lobes, and then a squarish shape versus a diamond with a median lobe. So what do these actually look like? So these are the extremes of the first 4 PCs. These are the actual outlines of those flagella. So PC1, you can see Columbiazomus, long rapier-like compared to Kenyazomus. PC2, Bamazomus, very spade-like with anterior-facing lobes compared to Schizomus without. PC3, Protoschizomus with these 2 posterior-facing lateral lobes. That one will come up later, compared to our friend Kenyazomus again. And finally, PC4 with lateral lobes and no median, compared to Cochin-Dolfarius with a long median lobe. So here's what the principal component analysis looks like. This is shape space or morphospace with all of our 283 outlines, translucent, so you can just see their density on PCs 1 and 2. All right, so what does this tell us? So first, we wanted to look at some of the differences in genera. So we have 64 genera, and this is all genera with a described male. And with this many groups, it is a mess, but it's a mess that shows there is a lot of overlap between the genera, and we have to drill down a bit more to get anything out of it. Then we looked at families. So there's 2 families, which are Protoschizomid in yellow and Hubbardeidae in purple. And you can see that the Protoschizomid, at least in these 2 PCs, is fully within the morphospace of the Hubbardeids. We looked at the morphospace, the biogeographic realm, and we saw that the neotropics is the largest of the convex holes. We also tried to code the Mexican transition zone, given how much work in Mexico is going on schizomid research. So we tried to determine or put a lot of care into whether a specimen was neotropical from the transition zone or Nearctic. And finally, we looked at habitat. Again, hypogean and epigean taxa largely overlapped in morphospace. So to try and quantify these differences, we looked at disparity, and disparity is a metric of phenotypic diversity in morphospace. In particular, for these, we looked at the sum of variance as our metric of disparity, which is the area occupied. So, of course, for genera, there is some overlap and some not. For families, we found that there was no difference in morphospace occupied. For realms, actually, yes. And in habitats, yes. So you can see on the right the box plots for the disparity for the areas that we looked at. These are colored a little bit by a number of taxa described in these areas. But I want to actually highlight for now the families being not different, given how different they seemed in our PCA. And it comes back to this Protoschizomus sprousei. And basically, if you look at only PCs 1 and 2 and not 3, you would get the impression that they are that the Protoschizomid morphospace is entirely within the Hubbardeiid morphospace, but that's not the case. And if you look at the box plots on the right, you can see if you're only looking at PCs 1 and 2, then there actually is a difference of disparity, but when PC3 is introduced, that goes away. So this scatter plot has PCs 1, 2, and 3 with the protoschizomids in red. And let's see if I can see my cursor. And here's the Protoschizomus sprowsi showing this big departure from the rest of the protoschizomids. So next we want to look at allometry. So is there a relationship between flagellum size or the flagellum shape and size? And for this, we, only 272 of the 283 outlines had associated length data. So PCs with variance of more than 1% from the total analyses were combined with the centroid sizes from the 272. And yes, we found that size is a predictor of shape. And on the right here is are the PC loadings if we did not control for size, that is, if size was not removed. If we didn't do that, then essentially size would dominate the plots at 80% of the variance. And to show that in a different way, the left is size corrected, which is what I showed before. And on the right actually includes size, so the small taxa are on the left and the larger taxa are on the right. But here we're only interested in shape and not size. So moving on, we wanted to look at how we can distinguish taxa using the Caribbean as an example.
That's 10 minutes.
10 minutes. Okay. So here, Cuba has the vast majority of the described species. And indeed, this purple plot for disparity, or the purple convex hull is Cuba, which is much larger than all of the others and actually encompasses many of the others. And while the small sample size in some did color some of the results, if you look at Hispaniola, which is the most comparable island in size, we did find a difference in disparity. We also found that disparity in island size are not correlated, and neither are diversity and island size. But if you did omit Trinidad and Tobago, you would get a relationship. So we suspect there's a relationship with the closeness to the continent. And finally, we wanted to look at whether flagellum size could, flagellum shape could help us distinguish genera. So these are the 12 genera that are found in Cuba. And in short, the large morphospace of Rolandius makes it hard to distinguish from most genera. But on the right here are the average shapes. So this could be another starting point for taxonomists and parataxonomists who are interested in distinguishing these genera. So this is the first time schizomid flagellum shape is analyzed morphometrically. The major axes of variation correspond to flagellum width, the widest point in pedicel length. There's an overlap in morphospace considering genus, realm, and habitat, but only realm and habitat have different hypervolumes. Flagellum shape changes allometrically. Neither diversity nor disparity in flagellum shape correlates to island area in the Caribbean. And dorsal shape of the flagellum can distinguish some, but not all genera found in Cuba. We're continuing this work by using CT scans, of which we have one of a schizomid, and we're hoping to apply that to that pipeline to what I've done in spiders. I just wanted to sneak a spider image in here. And I want to close by thanking those who helped me with these analyses, namely Erica Garcia and Chris Hamilton, as well as my funding, and you for listening. Thank you.
Well done, Bob. Good job. Our moderator dropped his connection. So that's me moving in now to take over for him. You did have a question here from Chewy. He wants to know, he says, great talk, Bob. Besides the outline, is it possible to incorporate the variation of dimples and grooves on the surface of these flagella?
So the Fourier analysis can only use outlines, but I am not, it does not play well with landmarks, but I think if we introduced the location of grooves and dimples perhaps as variables in a multivariate analysis. Perhaps we could find a relationship there.
And then we'll do one more here. Rodrigo Munjares, I can't see the whole name. Excellent talk, Bob. Did you include any member of Agastoschizomus?
Yes, we have Agastoschizomus in the protoschizomid group. Yes, we did.
And let's do one more here, since that was a short answer from Martin Ramirez. Sorry for the mispronunciation. Your initial images showed that there are pockets used for sex interaction. Are those represented?
No, because this is strictly based on outlines, but we can introduce those in subsequent analyses, and we're hoping to do that with that and the claw of the female, where that interacts with the flagellum of the male.
Okay. Thank you very much, Bob.
All right, thank you.